Welcome to our deep dive into the world of Vibrio species.
Always an interesting bunch, that's for sure.
We'll be using the second edition of Principles and Practice of Clinical Bacteriology by Gillespie and Hawkey to guide us.
Excellent resource.
As medical microbiologists, you know these bacteria can be quite a challenge. So we're here to give you the most important insights for diagnosing and managing those Vibrio infections.
Right, it's good to review.
Where should we start? What are the key characteristics? The ones that a medical microbiologist can't afford to forget.
Let's see, well, they're gram-negative bacteria.
Right, fundamental stuff.
And they're fermentative.
Okay.
And oxidase positive.
So that helps narrow things down a bit.
It does, yeah.
Now, what about their, lifestyle? What makes them unique?
Well, they're known for their motility thanks to their polar flagella.
So they're quite active under the microscope.
You could say that, yeah, like tiny speedboats.
Now, I've always associated Vibrio with the ocean. Is that a fair assumption?
It's a good starting point. They do thrive in the presence of sodium chloride, which hints at their connection to marine environments.
Right. Makes sense.
They belong to the Vibrionaceae family, which also includes Photobacterium, Aeromonas, and Plesiomonas.
So it's a family affair.
You could say that.
But just to clarify, are all Vibrios found in the ocean?
Ah, good question. Not necessarily. While many are found in marine environments, we do isolate some from clinical specimens.
So sometimes the lines get a bit blurry.
Exactly, especially when you consider the potential for contamination.
That's an important point to remember.
It is.
Now, we can't talk about Vibrio without discussing their ability to cause some pretty serious diseases.
Absolutely.
Everyone's heard of cholera, but what other kinds of infections do these Vibrio species cause?
Well, you're right. Cholera, caused by Vibrio cholerae, is the classic example.
Right.
But our source material in Table 34.1 actually lists a whole range of Vibrio species and the different diseases they cause.
It seems like a pretty extensive list.
It is.
So what are some of the other major players, so to speak?
Well, you have Vibrio parahemolyticus, a common culprit in food poisoning from contaminated seafood.
Ah, so that's one to watch out for.
And then there's Vibrio vulnificus, which can cause severe wound infections and even septicemia, particularly in people with weakened immune systems.
Yeah, those are the ones that can really cause some serious damage.
Absolutely. And the table goes on to list Vibrio alginolyticus linked to ear infections and a few others that can lead to various infections.
It's incredible how these tiny organisms can cause such a wide range of problems.
It really is.
Now let's delve a bit deeper into how they actually cause disease, the mechanisms behind their pathogenicity.
Okay, sure.
Each species likely has its own bag of tricks.
Oh, definitely.
But let's start with the most infamous one, Vibrio cholerae, and its potent cholera toxin.
Right. That's the one everyone knows.
When it comes to cholera, that toxin is the star of the show.
You could say that, and it's a pretty devious character. You know, it doesn't actually damage the intestinal cells directly, but it binds to them and disrupts the normal ion transport.
So it's more like a saboteur, disrupting a complex system from within.
That's a good way to put it. And the result is massive diarrhea and electrolyte imbalance, the hallmarks of cholera.
And that explains why cholera can be so devastating, leading to rapid dehydration.
Yes, exactly.
But cholera toxin isn't Vibrio cholerae's only weapon.
No, it's got more.
It also produces these structures called toxin co-regulated pili, or TCP for short.
Right, PCP, essentially they act as grappling hooks, allowing the bacteria to attach firmly to the intestinal lining.
So it's not just about producing the toxin, but also about securing a good position to deliver it.
Exactly, and there are even more toxins in its arsenal like ZOT and ACE that contribute to its overall virulence.
It's a complex system.
Very much so.
Now, what about those other Vibrio species we talked about? Do they employ similar strategies?
Many of them do, like Vibrio mimicus and Vibrio parahemolyticus. They often target the gut, disrupt normal function, and cause inflammation, but they have their own unique sets of toxins.
So even though the specific players may differ, the overall theme seems to be messing with the gut.
Yeah, that's a fair assessment.
Knowing how these bacteria cause disease, let's shift gears a bit and talk about how these infections actually manifest in patients.
Okay, sure.
What are the telltale signs? Which should clinicians be on the lookout for?
Well, the clinical features, as you might expect, vary depending on the species involved and the site of infection.
Makes sense.
Cholera, for example, as we've discussed, presents with profuse watery diarrhea, sometimes exceeding 20 liters per day.
Wow, that's an incredible amount of fluid loss.
It is,
Yeah.
and it can lead to very rapid dehydration, which is why prompt rehydration is absolutely crucial in managing those cases.
I imagine those other Vibrio infections present differently.
They do. You might see gastroenteritis with symptoms like nausea, vomiting, abdominal cramps, and diarrhea, but not as severe as cholera.
So a range of gastrointestinal distress, depending on the culprit.
Exactly. And then we have wound infections, especially after exposure to contaminated seawater. Those can be quite serious, particularly if Vibrio vulnificus is involved.
That's a good reminder to be careful around seawater, especially if you have any open wounds.
It is, and in some cases, especially in individuals with compromised immune systems, Vibrio infections can lead to septicemia, which, as you know, can be life-threatening.
So a wide spectrum of clinical presentations, from relatively mild gastroenteritis to potentially fatal septicemia.
Exactly, and that's why it's crucial for us as medical microbiologists to be able to accurately identify these culprits in the lab.
Speaking of the lab, let's move on to how we actually diagnose these Vibrio infections.
All right, good idea.
What are the key steps involved? What should we be thinking about?
Well, it all starts with proper specimen collection and transport. It might seem obvious, but it's essential to make sure those organisms stay viable for analysis.
So handling those samples with care is paramount. Now, what about the media used to isolate these bacteria? Are there any specific types that work best?
Standard lab media like blood agar can be used, but we often prefer specialized media like TCBS for its selectivity.
What makes TCBS so special?
Well, TCBS stands for thiosulfate citrate bile salts sucrose agar. Essentially, it contains ingredients that inhibit the growth of many bacteria, but allow Vibrio species to thrive.
So it's like a custom-designed environment for Vibrio.
You could say that, and here's a neat trick. Vibrio colonies that ferment sucrose, like Vibrio cholerae, actually form distinctive yellow colonies on this agar.
Oh, so that's a helpful visual cue.
It is, definitely.
But colony morphology alone can't tell the whole story, right?
No, you're right. We also use a series of biochemical tests, such as the oxidase test, to confirm we're dealing with a Vibrio species.
So multiple lines of evidence are needed to paint a clear picture.
Exactly. And don't forget about adding 1% ACL to most media. It's essential for the growth of most pathogenic Vibrios, except for Vibrio cholerae and Vibrio mimicus.
Oh, interesting. So those two are a bit different.
They are, and when it comes to Vibrio cholerae, serotyping is crucial to identify specific serogroups like O1 and O139.
Right, those are the ones associated with epidemic cholera.
They are.
So it's like we're piecing together a puzzle using different tools to reveal the identity of the culprit.
That's a good analogy.
But let's talk about speed for a second. Are there any rapid tests available for identifying Vibrio species?
Oh, there are. We can use microscopy for immobilization studies where we observe how the bacteria react with specific antisera. And we also have immunoassays and dipstick tests.
So those are particularly helpful for rapid diagnosis and surveillance.
They are, very much so.
It sounds like we have a whole toolbox of tests at our disposal, from traditional culture methods to these newer rapid techniques.
We do, and if we need to go even deeper, we can use advanced typing techniques like PFGE, MLEE, and MLST to characterize strains and investigate outbreaks.
Wow, it's incredible how much detail we can uncover about these microscopic organisms.
It is quite remarkable.
And all this knowledge helps us develop better strategies for prevention and control, which we'll delve into after a short break.
Okay, sounds good.
So stay tuned. We'll be right back. Okay, so that first question. Let's think about those key characteristics of Vibrio species.
Right.
We know they're gram-negative, oxidase positive, and motile with those polar flagella.
Right.
So that leaves us with option C, spore forming, as the one that doesn't fit.
You got Vibrio species are definitely not known for forming spores.
Right.
That's a survival strategy we see in other bacteria like Bacillus and Clostridium.
Yeah, helps them withstand those harsh environments.
But our Vibrio friends have their own ways of surviving, like forming biofilms and entering that VBNC state we talked about.
Exactly. Different strategies for different bacteria.
Okay, let's move on to question number 2.
All right.
Which Vibrio species is the culprit behind cholera?
Okay.
Is it Vibrio parahemolyticus, Vibrio vulnificus, Vibrio cholerae, or Vibrio alginolyticus?
This one's a classic.
It should be a familiar foe for any medical microbiologist. It is. The answer is C, Vibrio cholerae.
Of course, we've spent a good chunk of this deep dive exploring its pathogenic mechanisms and the impact of cholera.
It's a big one.
All right, ready for question 3?
Hit me with it.
Which toxin is responsible for that, massive diarrhea we see in cholera?
Okay.
Cholera toxin, Shiga toxin, diphtheria toxin, or tetanus toxin?
We're going back to the heart of cholera's pathogenesis here.
We are.
And the answer, the one causing those severe fluid losses, is A, cholera toxin.
Right, disrupting that ion transport in the intestine.
Exactly, leading to all that watery diarrhea.
And it does all that without even damaging the intestinal cells. It's remarkable.
It is a very subtle but effective mechanism.
Okay, for question 4, let's head back to the lab.
Back to the lab.
Which special agar is commonly used for isolating Vibrio species?
All right.
McConkey agar, blood agar, chocolate agar, or TCBS agar?
Think back to those colorful colonies we talked about.
I remember those.
The answer is D, TCBS agar.
It gives our Vibrio friends a growth advantage, doesn't it?
It does inhibits the growth of many other bacteria.
And those sucrose-fermenting Vibrio colonies, they really pop on that agar with that yellow color.
They do, very easy to spot.
Okay, last question. Make it a good one.
All right, let's see.
What's the primary treatment for cholera?
Okay.
Antibiotics, antimotility agents, rehydration therapy, or antitoxin therapy?
This one highlights a crucial point about managing this infection.
It does.
The most important thing, the cornerstone of cholera treatment, is C, rehydration therapy.
Got to address that dehydration.
Absolutely. While antibiotics have their place, rehydration is essential. It can be life-saving.
And just to reiterate, we need to avoid those antimotility agents.
Absolutely. They can actually make things worse.
Trapping those toxins in the gut, not good.
Not good at all.
So there you have it, 5 questions to test your Vibrio knowledge.
Hope everyone did well.
I think we've covered some really important information today.
We have a lot of ground.
From those tiny characteristics of Vibrio all the way to their global impact.
It's fascinating stuff.
But before we wrap up, I want to emphasize, these Vibrio species, they're remarkably adaptable.
They are.
We've seen how they can pop up in new areas, even cause new pandemics.
Like that Vibrio cholerae O139.
Exactly, a reminder that they're constantly evolving.
They are, always changing.
So, as medical microbiologists, we have to stay informed.
Absolutely.
New developments, resistance patterns, it's a constant race.
Knowledge is our best weapon.
Absolutely. Well said. And on that note, I think it's time to wrap up this deep dive.
It's been a pleasure.
I encourage everyone to keep exploring this field. There's always something new to discover.
Oh, definitely.
Until next time, stay curious.
We've talked about how to treat these Vibrio infections, but how can we prevent them in the first place?
That's a crucial question. It takes us beyond just treating the patient in front of us.
Exactly. It's about public health. We have to understand how these bacteria spread, what causes outbreaks. Then we can come up with ways to prevent them.
So it's about protecting entire populations, not just individuals.
Right. It involves public health measures like improving sanitation, making sure food is safe, and teaching people how to stay healthy.
We talked about vaccines for cholera. Are there any other specific ways to prevent it?
Vaccines are really important, but access to clean water and proper sanitation are absolutely essential too. Remember, Vibrio cholerae mainly spreads through contaminated water and food.
So we need to stop it from spreading, break that chain.
Exactly. If we can keep the bacteria out of the water supply and stop food from being contaminated, we'll see a huge drop in cholera cases.
I imagine public health education plays a big role too.
It does. People need to understand the risks. They need to know how important hand washing is. And they need to learn how to prepare and store food safely. It's about giving people the knowledge to protect themselves and their communities.
So it's a multi-pronged approach. Public health measures, education, and individual responsibility all play a part.
Absolutely. And it's an ongoing effort. We need to keep an eye on things, identify potential risks, and change our strategies as needed.
This has been a truly fascinating look into the world of Vibrio species, from their microscopic characteristics to their global impact. We've explored their complex ways of causing disease and discussed the challenges of diagnosing, treating, and preventing Vibrio infections.
We've covered a lot. It's a diverse group of bacteria that are constantly changing.
And I think we've given our fellow medical microbiologists a solid understanding of these intriguing and sometimes dangerous microbes.
I hope so. It's been great sharing what we know.
But remember, learning never stops. In microbiology, new discoveries are always being made. Stay curious, stay informed, and stay ahead of the curve.
That's right. The world of microbes is full of surprises. We have to be ready for anything.
Until next time, keep those microscopes focused and those minds sharp.